All right, good morning, everybody. Can you hear me? Looks like my microphone's working. I can't see anyone.
Yep.
Okay. Admit these people. All right, any questions before we begin? We have a lot to cover today. Exams due this evening, this night. I have office hours right after this, if anybody wants to talk about the exam. We have lab today on closed population capture recapture models. Okay, so big picture, we've been talking about estimating abundance of wildlife populations. We talked about census, population surveys, distance sampling, and then we moved into closed population methods for estimating abundance. There are a few models in the closed population models that date back to 1978 that we will cover, the Otis 1978 paper. And we're going over those models. The important thing we're going to talk about today is the statistical framework for writing out these models that we're using. And those are writing out mixture models. So essentially all models in wildlife ecology that have some type of complexity with them use mixture models. So the first half of the course we talked about generalized linear models and various types of generalized linear models, but we didn't explicitly account for detection probability. And now we're accounting for detection probability and estimating abundance. So I just posted lecture 13 slides right before class. Those are available now. And they deal with mixture models. And we'll cover, we'll talk more about mixture models in lab this week too, just to introduce the concept twice. I think it's important concept, and it's probably new to everyone, the types of methods we're talking about. So going over that repetition will hopefully help highlight some of the concepts that are important. All right, so let me share my screen. We are in lecture 13. We are talking about mixture models and what these mean. Mixture models essentially mean there's more than one possible outcome that can happen, and that outcome is going to determine which path we take in our model, if we go left or if we go right, and that depends. And sometimes it depends on things we don't know, but we can estimate. Okay, so starting off with an example. Here was a press release related to the Yellowstone grizzly bear population. They said population is stable. Well, how do they estimate what that population is? It's very hard to go out and count grizzly bears. Even things like distance sampling don't work very well, because when animals, if you're flying in a plane, if you fly over a forest, you can't see animals that are on the line. So we need some method for estimating the population size of grizzly bears, of really any wildlife population that's difficult to see. So how do we do that? Well, we talked a little bit about how they looked at population sizes in the past, not population sizes, but how they gained any information about wildlife before radio telemetry, before advanced techniques for sampling, when researchers went out and painted a bear, essentially. So I already showed those pictures. I normally wait for this slide, this lecture to show those pictures, talk about wildlife populations in the old days, but today's lecture is jam-packed and we have a lot to get through. So we've already talked about this, methods that we look at estimating abundance. That's the big picture. We're interested in estimating what big N is, what the population size of some wildlife population, because that's important for a lot of reasons. So we're on mark recapture methods now. We've talked about the other methods. And we talked about last time, the very simple mark recapture model was Lincoln-Peterson method, where we assume the number of animals we capture, Y of t, comes from a binomial distribution with some true population size N. We don't know N, but we want to estimate it. And detection probability P. We can't capture all individuals in when we go out and capture them, it's simply impossible. So we have to estimate about how many of the population we capture on average. So what's the capture probability P of an individual animal? So N is the unknown population size, and that's what we want to get at. Y of T is how many we capture in time T. Remember, we have 2 sampling events for the basic Lincoln-Peterson estimate, T equals 1 and T equals 2. M of T isn't shown in this model here, but it's how we estimate P and therefore N. And that's the number of captures in the second attempt that has a mark on it. And then P's are unknown detection probability we want to estimate. So we want to estimate both N and P in our simple Lincoln-Peterson method. All right, so how do they do that in the old days? These are the slides that look at the old way of capturing bears. We went through these pictures already. Kind of a unique history in wildlife ecology. You know, wildlife ecology, as a side note, is a relatively young field. The first book was written in 1933 on wildlife ecology. Anybody know the name of that and who the author was?
Leopold.
Yeah, exactly. Elder Leopold, father of wildlife management. And he wrote the book Game Management. In early days, wildlife science was based on studying game animals because people like to hunt them. A lot of our current model of wildlife management in North America, the North American model of wildlife conservation, was built around game management. And of course, that's just not true today. There are so many valuable wildlife populations to people that game animals still bring in a lot of the dollars for wildlife management, but we have a much broader spectrum in our conservation efforts. Okay, so 1933, wildlife management, or game management was the book. We talked about Lincoln-Peterson methods with our snail example where we capture maybe 6 animals, and if we have 15 animals in the population total, we mark those, we release them back, then we capture them again, and from those numbers that we get, we can estimate both abundance, detection probability, and uncertainty associated with those abundance estimates. Okay, I'm gonna mute someone here. Okay. All right, so Lincoln-Peterson method, very simple method for using marker capture data. Now, if we think about our Lincoln-Peterson method and how we actually translate that data to an Excel spreadsheet, it looks something like this. We have the individual snail that we capture, and if we mark those with individual identifiers, say a number instead of just a black mark, then we can keep track of the capture histories for each individual animal. We see the first 6 here were captured day 1, and on the second day, only 3 of those 6 were captured again. So that gives us a kind of a naive estimate of detection probability that when we release them, we capture about half of them in the second time. But we also capture 6 new animals in our second time, and that's going to help us estimate abundance. So we can use these numbers, y of t1, y of t2, m of t, to get n of n and p. So here are those calculations associated with that. Y of t1 equals 6, y of t2 equals 9 because we had 9 animals captured in our second event. There were 3 animals that were recaptured. Those are these 3 here because they were also captured in this first event. And with those numbers, we can get an estimate of 18. And then of course, our estimate is just an estimate. We don't know it exactly, so we want to put some uncertainty around that, and we can use that variance equation to estimate our 95% confidence intervals. All right, now we talked about Z a little bit in lab, and we're going to talk about it today in lab 2. But I want to formally introduce this latent variable Z now because this becomes really important. And this is kind of I'd say it's not really an abstract idea, but it's a new idea. There's going to be some variable Z that's going to represent truth, whether or not an animal is truly alive or truly dead. Now, we don't always know Z. Sometimes we know a little bit about Z. Like if we see an animal, we know it's truly alive. But we want to distinguish between whether we see an animal and whether the animal is truly alive or truly dead. We know if it's truly dead, we won't see it. because it died somewhere off by itself and nobody may ever see it again. So we're going to represent y are going to be our data, what we actually observe, just like we always have in this course. But now we're introducing this concept of a latent variable called z, which represents some unknown truth or some partially known truth. And this is going to describe our mixture models. All right, so here's a simple example for this latent variable z. So imagine we have our garden experiment where we're trying to estimate the number of snails in a garden. Now, if an animal was truly there, then we say zi equals 1. And if we saw it, given it was truly there, yit is going to equal 1. So yit represent our data that we actually observe. Zit aren't always observed. However, if we do see it, if we do see the animal, if yit equals 1, we know that it was truly alive. This is assuming we're not counting dead animals, which you rarely see. And sometimes you see dead animals on the side of the road, but in our study designs, we usually don't develop surveys like that. Okay, so we're going out looking for snails. We're only counting live animals. If we see it and it's alive, we know that it's alive because we saw it and we saw it to be alive. But there's another case too. Actually, there's 3 different scenarios that can happen. This is the first scenario, and this one's the easiest. This is when we know the most information, when we actually see an animal. There's another one. It could have truly been there and alive, but we just didn't see it. And remember, why are our data, whether or not we observe it? Zit is some true state that we don't always know. So when we don't see it, we don't know what Zi is for this animal at this point. We may know later, which I'll get into, what Zi was. But all we know is we didn't see it, and therefore, if we didn't see it, it could have either been alive or it could have been dead. And if it was alive, Zi is going to equal 1, and if it was dead, Zi is going to equal 0, which is our third possibility. So the third possibility is it was not there, Zi equals 0, and therefore we could not see it, so Yi has to equal 0 with probability 1. So in all 3 of these outcomes, we only know Z when we actually see the animal. And then we don't know what Z is when we don't see the animal, but we can try and estimate what it is. So, all right. If we did a third survey and we didn't see this animal, this snail, in our second survey, but we went out and saw it in our third survey, we would know that for our last survey, Zi really equaled 1 because it had to be alive in the last period to be alive in the next period. So from these types of data, this is how we can estimate what Zi really was. And there are going to be some cases where we don't know exactly what Zi is going to be. And those cases are when animals are seen once, they're marked, we know that they're alive then, but then we never see it again. We're not going to know if it died at all, if it was alive the whole time and we just never saw it, or if it actually died in one of our sampling events. And in those cases, we don't know what Zi is. But if we have enough samples, we can estimate some probability based on the Zi's that we do know of survival. All right, so here are those 3 possible outcomes and what they look like for our data. Remember, our data are what we see. And if we saw it, if it was there and we saw it, Yit equals 1, and then we know Zi equals 1. Notice I'm distinguishing the subscripts on here. These are closed population models, meaning, yeah, I want to make a point of clarification here because I've been blending 2 scenarios. These are closed population models, meaning we're assuming they all stay alive. There are no births, deaths, immigration, or emigration in these examples. And the way I was describing up until now was using our open population models. So let me reframe this. The animals are either going to be alive or dead in our study. We're assuming there's no deaths and births, immigration or emigration. So Zi is going to be fixed. They're either going to be in our study area or not in our study area. Zi is going to be 1 or 0. And that's not going to change. So I represents the individual snail. But we're going to go sample many times. And that's why we have YIT subscripts on our actual data. So it can either be in our study area or not. If it's in our study area, Zi is going to equal 1. If it's not in our study area, Zi equals 0. So let me just go back to clarify exactly for our closed populations what Zi means. It was truly in our study area, Zi equals 1. And then if we see it, YIT equals 1. If it was truly in our study area, but we didn't see it, Zi equals 1, YIT equals 0. But if it was not in our study area, Zi equals 0, and therefore we could not see it, YIT equals 0. When we switch to open population models, Zi will represent births and deaths. Right now, it just means if it's in our study area or not. Any questions about that? Okay, I want to
Yeah,
focus.
I have a question.
Yeah, go ahead, Leela.
Would Zi equal 1 and Yi equal 0 only in the second time that you look at it? It could never do that for the first time, right? Like the first time you go out.
The first time you go out, if it was alive and you didn't see it, YIT would equal 0, and Zi would equal 1.
But then how would you know that it was in the study area?
How would you know it was in the study area? If you didn't see it, you may see it in the next time, period.
Perry, is Z like probability where you can't necessarily like observe that? It's just kind of a fact about the system?
Yeah, good question.
Okay.
And this is why this is new. So it's not a probability, it's a state of nature. It's either yes or no. There's some truth, either 0 or 1, but we don't necessarily know it. So if we were some oracle, this is how some people in stats conference talk about this concept. If we were some type of oracle and we knew exactly what the state of nature is, we could look through our looking glass or whatever and say, okay, this one's alive, Zi equals 1. This one's outside of the nature. Now let's see if these silly surveyors out here actually detect whether or not it's alive or not. So Zi, you can think of as some true state of nature, but it's unknown. The animal's either alive or it's either in the study area or it's not in the study area, but we don't necessarily know that. We know it in some cases, only when we see it. If we see it some point in our study, we will know it's in our study area. So this gets back to Leela's question. If you go out there and you don't see it, can Zi equal 1 and YIT equal 0? Yes, if you go out the first time and you don't see it, but you go out the second time and you do see it, you knew Zi equaled 1 because we're assuming no emigration or immigration, a closed population. So we knew it had to be there the first time. But YI1, the YIT for time period 1 equals 0 because we didn't see it. YIT for time period 2, T equals 2, equals 1 because we did see it. And in that case, we knew that it was in our study area because we're assuming this closure assumption. We just didn't see it the first time. That's how we're going to estimate detection probability. Because we're going to see animals in our second survey that we didn't see in our first survey that we knew were there because it's how we designed the survey. And that'll tell us something about how many we missed. All right, any other questions? This is a new concept. So I'm happy
We
there
don't
are.
use Zi in any of the actual stats that we're doing, right? Like in any of the equations, because we can't actually know what it is. Or we can only prove that something's there. We can't like prove that something isn't there.
We can estimate the probability of Zi being a 0 or a 1, just like we can estimate the probability of an animal that's not there that we don't see. So when we think about detection probability, it's kind of the exact same idea, right? We know that there are animals out there that we just don't see. And that's why we try and estimate how many of them we don't see. It's the same exact concept. There's animals out there that we know we're not seeing, that zi equals 1. And we want to estimate how many of those are actually there. When it comes to fitting the models, we won't see ZIT. It's done in the background. But it's important to be able to write out what's going on when R's fitting these models so that you can interpret it related to the biology of the system. Okay. All right, so here are 3 possible outcomes that can occur. And when we have these different outcomes, then we use a mixture model. So we don't always know what ZI is. If we were some oracle, we could see what ZI was. But in absence of that power, we have to estimate how many that we actually don't see that were truly there. If we saw it, we know that it was there. So that's why we call this a partially known latent variable. Sometimes we know what ZI is, sometimes we don't. If we didn't see it, YIT equals 0, it could have been there and we just missed it, so ZI could equal 1, or it might not have been there at all. ZI could have been 0. So we want to estimate how many are truly there that we missed. This is the number that we're going for. This is the difference between how many we actually see in all of our surveys and how many might have been there that we didn't see during any of our surveys. So N, big N, is the sum of all of the ZIs. That's how we get to our big N. What's the population size? This is the sum of all of our ZIs for all of our individuals. So let's look at our snail IDs here, our snail example, and how we would write this out in an Excel table. Here's our Ys. In the first trip, we captured 6. In our second trip, we captured 1, 2, 3, 4, 5, 6, 7, 8, 9. 3 of those were recaptures. If we were some oracle, we would know that there were 16 total there, but we only saw 12 of them. So we have to somehow estimate the remaining 4 that were there that were never seen in any of our surveys. Okay. So just as a quick exercise to try and understand these concepts, let's look at filling in these question marks here. Take a second and fill in as best you can the unknown Z's here. Remember, these are partially known. We know some of them, we don't know any of them. We don't know all of them. There's 4 values we need to fill in here, just as a self-check. Okay. So let me call on someone I haven't heard from in a while. Matthew, what do you think?
So would the first one be 1?
Yep.
The second, we don't know. And then the third would be 1. And then the fourth would be 0.
let's see. Okay. The first one you said was 1. That's correct. We saw it. We saw it. We know it was alive. The second one you said 0. So when it says 0, what does that mean? So this is all brand new today. So keep that in mind. You said it would be 0.
I said we wouldn't know.
We wouldn't know. Okay. Okay. So let me clarify one concept here. We saw it on day 1. We knew it was alive because we saw it on day 1. Or we knew it was in our study area, sorry, on day 1. And remember, the assumption was we have a closed population, meaning if it was there, it had to be there throughout the remainder of the study. So because we saw it on day 1, we know that it was in our study area, and therefore the second one would be a 1, too. Follow me?
Yeah.
Okay, yeah. All right, how about the third one you said was a 1, correct?
Yeah.
And the fourth one you said was a 0. Okay.
Yeah.
So why do you think that one was a 0?
Just because we didn't see it either time.
Okay, right. So we didn't see it either time. So one of the possibilities was we didn't see it, and therefore it wasn't there. But then remember, there's 2 possibilities that can happen when we don't see it. It could be that we saw it and it wasn't there, and then it would be a 0. Or it could be we saw it, it was there, we just didn't see it, or we failed to detect it in both of those cases. So the fourth one is the one we truly don't know when we have a closed population. So the fourth one is a question mark. We didn't see it, so we know very little about it. It could have been there and we just didn't see it, which in case Z would be a 1. But it also could just not have been in our study area. And therefore we would never see it, no matter how hard we looked. So it's that fourth one that we don't know. The first 3 we know because we saw it at least once at some point in time. So the answer is 1, 1, 1, 0. Any other questions about that? Thank you, Matthew.
Yeah, if you go back to the previous slide, 15 and 16 both have 0 and 0, but then the unknown truth is 1.
Yeah. So this is, in this case, yeah, that's a good point. Let me clarify. I see where that was confusing. This is truth. We're an oracle, we can see the truth. So we know that there were 16. We saw in our first slides here that there were 16 snails. Yeah, I have 16 in here. There are 16 snails in this population. So this slide right here represents how we would actually write this out if we knew what truth was. But when we don't have this oracle power, right? We don't know how many there actually are in there. If these are the data we see, the exercise is, how would we fill out this ZI information? So the difference between this last line here, 0, 0 question mark, and the previous slide, 0, 0, 1, is this shows what that scenario would look like in truth. And this shows what us as observers would actually see, or what part of these Z's we would not know. Okay. Any other questions about that? I'm glad we made that point of clarification. All right, so recall back to our Lincoln-Peterson method where we had Y, T were how many we counted, N and P are what we're interested in estimating. Now we have information on each unique individual. With our Lincoln-Peterson method, we didn't necessarily mark each snail with a unique number. we just marked them with a dot, some way that would tell us that it had been captured before. But now when we have unique information on each individual eye, we learn more about the population. We have more information. So we can use this information in our model to get better estimates of closed populations. All right, so Lincoln-Peterson method, the assumptions were the population is closed so that n is constant. We don't have any subscripts for t on n, meaning it doesn't change through time. There's just one fixed value of n. There's no immigration, emigration, births, or deaths. This is assumption Lincoln-Peterson method. It's also an assumption of closed population capture-recapture models, which we're talking about now. And we had individuals do not lose marks between two sampling periods. All individuals have the same probability of being captured. There's no I subscript on P. We can change this when we talk about behavior models, which we'll do in a second. Marking animals does not affect catching them, so there's no behavioral reaction to traps, and the probability of capturing animals does not change in time. So the closed population capture-recapture methods allow us to take the Lincoln-Peterson method and relax some of these assumptions by collecting individual information on animals. Specifically, those assumptions that we're going to relax are 3 to 5. This assumption is the assumption we'll relax when we move to open population mark recapture methods. So we start off with Lincoln-Peterson method. It has the most strict assumptions, but it's also one of the simplest to do. The methods for calculating abundance are very straightforward. All we have to do is capture two populations in a row. And capture I use generally. And that can mean a lot of different things. And as long as the population is closed, we can estimate abundance, and if we meet these assumptions. However, often in studies we can't meet all of these assumptions, so we want to relax those methods, and we're going to use these closed population capture-recapture methods to do that. Now we get into mixture models and how we write out the closed population capture-recaptures. When we see this case here, this is like, if Zi equals one thing, we're going to do one thing, and if Zi equals another thing, we're going to do another thing. So it's like a dichotomous key almost, and it depends on what Zi is. If the animal is truly not in our study area, then YIT equals 0. However, if it's truly in our study area, YIT can be either a 0 or 1, meaning we can see it or not see it. And that's going to depend on our detection probability, P. So let me start at the beginning here. Here is our first mixture model. YIT are our data. This is an Excel table that has rows for each individual I and columns for each time period T. So any cell YIT in that data can be modeled in the following way here. It's going to depend on our detection probability P. We don't know what P is, we want to estimate it. It's also going to depend on whether the animal was truly in our study area, Zi equals 1, or if it was not in our study area, if Zi equals 0. Zi is whether or not the animal is in our study area. We don't always know Zi. We only know Zi if we've seen the animal in our study area, and if we've seen it, we can say Zi equals 1. If we haven't seen it, we don't know. And that's to say, we don't know how many animals are in our study area that we have never seen. That's what we want to estimate. How many animals within our study area that we have never seen. We know there's some of them that are good hiders, and we want to try and estimate how many there are. And we can do that by estimating what P is. Any questions about this? Okay. If you do have a question, I'm sure you're not the only one with that same question. So please feel free to ask. These are new concepts. All right, now Zi is something we don't always know. And what we want to do is estimate a parameter using the data, the zi's that we do know, to try and estimate psi I. That's the probability of an animal being in our study area. And that's going to tell us about how many animals are in our study area that we never see. Combination of psi I and p. So p is the probability of observing an animal given it's in our study area. And psi I is the probability of an animal being in our study area. When we get to open population models, when animals can actually die within our study period, we're going to use this exact same framework, but it's going to be slightly in different interpretation. And I'm just going to say what that interpretation is now, because maybe it'll help conceptualize what psi I is. When we get to open population models, YIT is going to be the same thing it is now, whether we observe the animal in animal I at time t. And that's going to depend on detection probability. But the meaning of ZI is going to change. It's going to change from whether or not an animal is truly in our study area to whether an animal is truly alive or not. And if ZI equals 1, it's truly alive. And if ZI equals 0, it's truly dead. And then psi I is not going to be the probability of an animal being in our study area like it is for closed population capture-recapture models. But in open population capture-recapture models, psi I is going to be the survival probability, the probability of one animal surviving from the last time period to the next time period. So we'll get to that when we get to open population models. But for now, ZI is whether or not the animal is in our study area, and psi I is the probability that an animal is in our study area. So again, YIT, if the ith animal was seen during the tth survey. P is the detection probability that we need to estimate. We don't know what this is, but we have data Ys that are going to help us estimate that. How do we estimate P? Well, kind of naively, we're going to see some animals in the second one that we're not going to see in the first one, and we're going to see some animals in the first survey that we're not going to see in the second survey. And by kind of comparing those relative numbers, that'll tell us something about the probability that we actually observe those animals. ZI is a partially known latent variable describing whether an animal was truly in the study area or not. And psi I is a probability an animal that wasn't seen was in the population. For now, this is somewhat arbitrary. It's going to help us estimate how many Z's that we didn't see. But psi I becomes really important when we talk about open population models for the reason I just mentioned, that it's going to represent the survival probability. We're introducing it now, so we see how this framework, this mixture model framework, underlies all of more complex wildlife population models. All right, so here's an example. We're going to do 3 surveyors. They're going to go out and look for nests in the field. So this is related somewhat to the lab data, the test, or the chicken egg data that we talked about. We have 18 nests that are truly out here. So if we count all these, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 nests. We're going to have 3 capture occasions. We'll have 3 different observers go out and try and find nests. There are 1, 2, 3, 4, 5 of these not in our study area, and the rest of them are in our study area. So the truth, if we were some oracle and we could look at this plot like we are now, we see that 5 of them are not in our study area. That represents these 5. And the other 13 of them are in their study area. So this again describes the difference between the interpretations of those graphs that Leela asked about. There's some truth that's fully known, and there's some truth that we try and fill in what we can know. All right, so we have first observer here. He walks through the study area and he finds 4 nests, finds this nest, labels it nest 1, finds this nest, labels it nest 2, 3, 4. All these other nests are in here, but they weren't observed. So he marks the location of these nests, and those ones get a 1 in our Y values. So our Ys, they were seen by the first observer, 1, 1, 1, 1. We know the truth here. We can fill these in because we saw it. We know it's in our study area. And we don't know anything about the rest of these observations. So we're gonna have to go back out and survey again. All right, so now this observer goes out. They find 3 nests. They find the first nest that observer 1 found. They find the third nest that observer 1 found, but they also find an additional nest here, the 5th nest. So we see we can fill in our data here. Of the first 4 that observer 1 found, the first and the third nest were found the second time, and the second and the fourth nest weren't found, and they found 1 additional nest. So we can fill this in. So we have 0 and 1. Now we know the truth for these ones. So here's the known truth. And we know partially known truth here. The known truth that we know just because we get to see the big picture of everything, the oracle view. And the data that we actually observe and collect. So now we're starting to learn something about detection probability from these 2 values here. We know that there was 1 nest that was observed the first time and it wasn't observed the second time. So we know we're not seeing all the nests. And we're going to be able in the end to combine these columns to estimate our detection probability P. Same here, we didn't find this first nest in the first go around, and we did find it the second go around. All right, so our final observer, we have, the final observer goes around, finds a new nest in 6, 7, and 8. I think I made this example last year when Game of Thrones was just wrapping up. So we have Jon Snow flying on Ragnar, looking for nests. Anyway, they find 3 new nests here that weren't observed. So with that, we can fill in our data. We have our third capture probability. A third observer found the three new nests here, but they didn't find any of the other ones. We know that there are 1, 2, 3, 4, 5, 6, 7, 8 nests out there because we've seen at least 8 nests in our 3 surveys. And what we don't know are all of these ones down here. How many are there? Actually, these ones here that are actually in our survey area that we never saw. That's the number we're trying to get at. Okay, yeah, so these ones right here. So here is model M0, and we'll talk about this in lab today for those who haven't seen it. This is, we're assuming the detection probability is the same for each of our 3 observers. They all had the same probability of observing a nest given it was in our study area. Remember, if it wasn't in our study area, if Zi equals 0, those are the nests that are out here. And the person's not surveying out here, so there's 0 probability of finding those nests. We don't care about those ones really right now. What we care about are the numbers that are in our survey area that we didn't see. Nests 9 through 13. Because we want to know about how many are in our study area. And a more flexible capture-recapture model than Lincoln-Peterson model is our model M0. And this allows us to estimate our detection probability and how many Z's are actually within our study area. Okay. So there's our very basic model, so we have our Lincoln-Peterson model. That is I'd say that's our very basic model, Lincoln-Peterson model. We don't have to individually mark nests or animals. We can just say how many had marks, how many didn't mark. And what I mean by individually mark is we mark them, but they don't have unique identifiers to that specific animal. We don't know nest 1 from nest 5. We just say, here's a nest, and we captured this one last time. A little more complex than that is model M0, which now we're keeping track of individual animals or individual nests, and we have a unique capture history. These are our capture histories for each individual. And model M0 is the basic type of those models where we keep track of each individual with these I subscripts. And model M0 says the detection probability P is the same no matter what day you go out and no matter who's the surveyor. Okay, so we just have one detection probability. Now, if we go back to our model selection procedure when we were doing generalized linear regression, we also had a basic model. That was our null model, basically an intercept only model where we only had beta 0 in our parameters. This is the equivalent to that model. This is like our null model. That's why we have the M0 here as our null model. It assumes that detection probability is constant for each person, for each survey. But we can add some more complexity to our model in the same way we did for generalized linear regression by adding covariates to our surveys, adding more parameters, and saying, well, maybe P isn't constant for each person that goes out and surveys. Maybe we want P to change depending on who's surveying or what the weather is. So now we're going to add complexity to this basic model M0. M0 again assumes that our detection probability P is constant for each surveyor. But let's say we have 3 different observers. They all have very different ability to find nests, and we want to estimate a different detection probability for each individual. And that's our next more complex model. That's model MT. So model T says we're going to change the detection probability for each time period that we're surveying. So we have 3 different observers here in our time period. We want to estimate a detection probability, one for, I forgot his name, Zissou from Life Aquatic. And then we have another detection probability for time period 2, our oatmeal cartoon. And then we have Jon Snow from Game of Thrones. Okay, so we have 3 different observers and we think they all have different ability to find nests. So let's change P for each one and estimate it separately. Well, the way we do that is very similar to the framework we've already established for generalized linear models. We have P of T, which has to be between 0 and 1 because it's a detection probability. And we can assume that the logit of P of T is associated with these covariates. Beta 0 represents our baseline P. That is for the Life Aquatic. Our second one is our oatmeal cartoon, our surveyor. So if beta 0 plus beta 1 is going to be the log odds of the detection probability for that observer. And then our third one will be beta 0 plus beta 1, which will be our detection probability for Rhaegal and Jon Snow surveying. Okay. So we can make our models more complex in the exact same way we did with when we saw in our generalized linear models with our Bernoulli regression we did before. So we're using the same framework we've already developed and learned, but now applying it to more complex models that allow us to deal with detection probability. So in this case, YIT, if the ith animal was seen during the tth survey, that's the same. P of T, now notice P of T here has a subscript on it. That means it can change for each time period. In our previous example, in our null model, P did not have a subscript. There was no T associated with it. So it was going to be the same no matter what time period we were in. Now it has a subscript, the time-specific detection probability for each observer that we need to estimate. And again, Zi is the partially known latent variable describing whether an animal was truly there or not. And psi is the probability an animal that wasn't seen was in the population. Okay. Now, we can keep adding different scenarios to all of these models. We started off with our basic model M0, which assumes detection probability was the same. We can allow detection probability to change for each time period or for each observer. And we can allow detection probability to change based on other factors in the environment. And that may have to do with habitat availability, habitat choice, that's the habitat conditions that are in each location, and other things. So we have MH, our heterogeneity model, which allows detection probability to change for each nest that we observe. Now our models start to get a little more complex. we have a P, the subscript on P is I this time, meaning P can be different for each individual nest I. We have the logit of PI has some overall constant value, beta 0, so this is kind of like model M0, in that it has one overall value, but it has some residual error on it. So this is exactly like we saw when we were talking about residuals with normal linear regression model. And residuals are just a little bit, are the difference between beta 0 and individual I. Now we assume our residuals, just like we did before, come from a normal distribution with mean 0 and variance sigma squared. So this model actually has two parameters that we're estimating. It has the parameter beta 0, the overall detection probability, and it has some estimate of variance that describes how much different the detection probability is from each nest to the next nest. And if you have drastically different conditions for each nest, then sigma squared might be large. But if they're all pretty similar, sigma squared might be very small. All right, same interpretation as we had before, except PI's nest-specific detection probability we need to estimate. MB, this is a behavior model. So now, the first observer goes through a field, finds a nest, and after they find the nest, the bird says, well, I'm getting disturbed too much here. You detected me, so I'm out of here. This was a bad place to put a nest. Someone came by, they left their scent all over, and I'm gonna leave. I'm gonna abandon this nest. Maybe I'll take my eggs with me, maybe I won't, and go somewhere else. And you'll never find that nest again. So that's a behavioral response. This has to do with trap happy, trap shyness of being detected. And the model looks like this, and we'll talk about this in lab 2, where we have our detection probability P, but P is going to depend on whether or not that nest has already been found. And if it's been found, and when the nest is found, that decreases the probability that you'll find it again. Beta 1 is going to be negative. It means if it's going to be found, the likelihood of detecting it again is going to decrease. In some cases, after you catch an animal, they're more likely to be captured. We see this with rodents and Sherman traps if there's a food reward inside the trap. So if it gets trapped, finds some peanut butter in the trap, and then likes the peanut butter and comes back again and again, we see beta 1 is going to be positive. So it's going to increase the likelihood of an animal being detected given some behavioral response. So now we have two parameters to estimate: beta 0, probability of being captured the first time, and then beta 1 is the offset for whether or not it's different to be captured the first time versus the second time. Okay, now there's a lot of methods. I'll start off with this next time. I'm 3 minutes over. We'll start off with this video on removal methods, but a lot of times when you you do sampling, you don't always put the animals back where you found them. We see this with invasive species and so on. And the way we modify our models for that is we just set the probability of recapture to be 0. So beta 1 becomes very negative. If the probability of being recaptured is 0, then we can adjust that in our model. Okay, so I have office hours now and I'm happy to answer any questions.